So thank you for the invitation to come and speak to this group tonight. I'm really excited to share the work that my lab, which is mainly undergraduate students at Roger Williams University, has been working on for the past few years now. Um, I hope that what I do tonight is convince you why the coral that we're working on that is here in Rhode Island is worthy of state emblem status. So as Abigail mentioned, this is an effort that's been ongoing now for a little bit. It actually started before the pandemic, got put on hold, and I'm thrilled to say that now things are moving forward up in the State House, and we're working to designate the northern star coral as the official state coral of Rhode Island. We'd be the first state to have a state coral. And what I want to show you today is some of the research and innovation that's being done by students here in Rhode Island, a little bit about the history of research on Astrangia in the state of Rhode Island, which is actually where it's deeply rooted. And then sort of, I think, go into some stories about how this coral has the potential to really reveal a lot of secrets to us about how other corals across the world function, how they work, and maybe even some tips into improving the health and survival and outcomes of tropical corals that are facing uh, all of the threats associated with global climate change. So with that, what I want to do is sort of start off, I'll just warn you now, I'm going to start off with the bad news. And then what I'd like to do is sort of provide you with a few stories that show you why we have some good news. I believe it's really important to get the bad news out of the way first. I think what that does is it's important to tell the stories that give hope, right? That give people something to hang on to and give us something to say, you know, well, what can we do? You know, yes, there are problems and we're aware, but what is it that we can do to address those problems? And so the bad news is this, is that there are a lot of different international agencies who look and collect the data and have agreed that coral reefs across the globe are in great trouble. And the current estimates are that by the year 2050, nearly 3/4 of the world's coral reefs as we know them will likely be gone. And it may actually be moving more quickly than that estimate. The past few years have been pretty grim for coral reefs on a global scale. This is because of global climate change and human-induced ocean warming. The main threat is what we call coral bleaching. So if you look at this image that was taken from the Catlin Sea View survey through the Ocean Agency, this is run by Richard Beavers, what we can see is a shot of a reef in the Pacific in 2014 on the left-hand side. And if you look at that image, what you likely see is that there's a golden brown hue to those corals. And those corals are, it sort of looks like a thicket of branches. That golden brown coral is due to an algal symbiont, so a partner that basically lives inside of the tissues and uses light to produce sugars just like plants do. And that's what feeds the coral animal. And so, yes, if there are some of you out there who are surprised that corals are indeed animals, this thing that looks like a big thicket of maybe some kind of plant, it is indeed an animal. And I'll get some close-ups to show that to you. So when temperatures heat up, as they did, as they do every year, we had a major bleaching event in 2015. And what happens when temperature heats up is that corals basically have this partnership with algae that live inside of them. And when temperatures get too high, they basically kick out those algae. And that doesn't necessarily kill them unless the warming lasts for long enough that they never get the chance to bring new algae back in.
I can't deal with that.
And and so if they don't acquire new partners, if they don't bring in new algae, then they will die from starvation, basically, because they don't have partners providing food to keep them alive. And so this is indeed what's happened in this progression here. By the end of 2015, this particular thicket, instead of regaining algae and that sort of beautiful bronzy glow, what this is dead coral skeleton that's been overgrown by algae, by a different kind of algae, a sort of turfy, messy algae that does not keep the coral alive. No, sometimes they don't die from starvation, but in bleached states, they're vulnerable to infection by other microbes like fungus or bacteria. And years of research on coral shows that often disease is not due to infection by a single pathogen, like what we think of as, you catch a cold, it's a single pathogen. But more commonly in corals, what we see is that there's a community-wide shift. So overall, the entire microbiome makeup is somehow altered. And so corals are really complex because they're sitting in an ocean full of water, right? In seawater, there are literally millions of cells for every milliliter of seawater, and they're bathing in that, millions of bacterial cells. And so they have to be able to prevent infection by the, for, to simplify it, but infection by the bad guys, right? And be able to promote the growth of the good guys, right? So there are some bacteria that can protect them and provide benefit to them. But when things get off kilter and we don't always understand what it is that leads to these kinds of diseases, but typically it's things like increased temperature, nutrient runoff from areas with high levels of fertilizer, um, increased UV stress, maybe loading of sedimentation. Basically, what happens is we see these community-wide shifts, and we know enough now to be able to say, if we look at what bacteria are in a sample, we can say, oh, that's a disease signature. And the same goes for human health. In fact, a lot of this might sound familiar to you. Um, the gut microbiome has received a lot of attention in recent years for good reason. It's so similar to human health, this story. And really, from what we know about human health, we know that the gut microbiome makeup is linked to so many aspects, right? A disturbed gut microbiome can impact everything from digestion to perhaps neurological health. And a lot of clinicians have shown that a disturbance in the gut microbiome has been linked to diabetes, to anxiety, to depression, dementia, Alzheimer's. Um, and now in humans, it's a very common practice as part of preventative health. People eat yogurt or other fermented foods to bolster their gut health, to get the good bacteria in, right? And another level of that is a diet that's high in fiber, right? Doctors recommend low in saturated fats, high in fiber, and that's shown to boost the growth of good beneficial bacteria. And it's shown to reduce the growth by harmful bacteria. And if you take antibiotics, perhaps you do sort of a double dose of yogurt, right? And things like antibiotics and diets that are high in saturated fats have been suggested to cause a disturbance. And that is what microbiologists call dysbiosis. It's basically a disturbance in this symbiosis between bacteria and human, right? Or in the case of a coral, bacteria and coral. And so there are scientists now that are actually doing really brilliant work applying probiotics. So they've made custom coral probiotics, and they're applying them now in pastes and injecting pastes onto diseased corals. And on a very limited scale right now, they're just starting to show that they can cure corals from a disease that's ravaging the Caribbean using this technique. So the problem that I've been really fascinated with for the past 10 years is how can we make sure that corals can really recover from a disturbance? It's impossible in tropical corals to study recovery from a disturbance, such as bleaching, without first disturbing them so much that we stress them, where we heighten their immune system, we change everything that's going on. And so it's really impossible in tropical corals to get corals without algae, without hurting them. And that confounds the experiment, let alone it uh, kills corals that already are threatened anyway. So it becomes really problematic. And so about 10 years ago, I was introduced to a new system where algae can be functionally absent. And so this is where the good news is. So our local coral here in Rhode Island looks like this. This is an underwater shot from a site nearby. And this is a Strangia poculata, the northern star coral. And there are a lot of things about this coral that make it really unique. So it's similar to tropical corals in many ways. It's in the same group on the tree of life. It's a cousin to the jellyfish and the anemones. Um, And it's different in really, really important ways. And because of that, it's a great model for understanding tropical corals and their response to climate change. So I'll tell you a few of the reasons that it's so uh, that what lends itself to being a great model for studying corals. And the first is that it also has an algal symbiont, just like tropical corals do, but it doesn't need it. So if we look at these corals underwater, which is what this picture is here on the left, some of them have that golden brown color from algae in their tissues, and some of them are just stark white. They actually do have a few cells of algae in them, but they're very, very low, so low that they don't really make a contribution to the host. And so up here, those of you who have maybe been diving or snorkeling in tropical waters, you know that down there it's very clear. There's not a whole lot of stuff floating around in the waters. And up here, there's a lot more, um, you know, particles and things in the seawater, things that corals can actually capture and eat. And so this coral has evolved to be an aggressive feeder. And because of that, it doesn't need its algae to survive. And so we can see this in basically it's some colonies that are symbiotic. They're sort of a dark chocolate brown. We have some that are what we call aposymbiotic. They basically lack or have very, very low levels of that algal partner. And then some that are sort of in an intermediate state, or what we call mixed, where they seem kind of patchy. So that's the most important way that Astrangia is different than its tropical cousins. It's pretty flexible in that partnership, and that gives us a naturally occurring experiment where we can say, with white corals and with brown corals, if we do something to them in the lab, say treat them with different bacteria, how do they respond differently? And there's a lot of power in those experiments. The other thing that's really important, the article that you mentioned, Abigail, mentioned this as well. I like to call these corals New England tough. So they can handle our really, really cold winters. And I am someone who spent most of my adult life living in the tropics, so I'm especially sensitive to this. And I find it fascinating that there are both people and organisms that can do this. I'm still working on it myself. Um, but what you might be able to see in this picture, these are some of our divers going in the water. There's actually snow falling, and this is an April collection, I think in 2016. And they were able to get plenty of corals during that snowy, cold day. But then I personally really enjoy collecting corals in the summer months as well. And the corals do well and they survive year round these extreme temperature fluctuations. The other thing that's really striking about Astrangia that's very different than its tropical cousins is that it has a really large range. And so if you look at the different dots on this map, each one of these dots represents a place where Astrangia has been either spotted or collected. And so all the way down off the coast of South America, and then up to southeastern New England. And we're at sort of the northernmost range here in Buzzards Bay. And so again, because of that, there are different populations that are experiencing, the ones down toward the equator are experiencing very, very little fluctuation throughout the year. And then up here, extreme differences in temperature throughout the year. So because of those different factors, this coral really lends itself to experimentation that can reveal a lot about coral physiology that just simply are experiments we can't do in tropical corals. And so what I'd like to tell you about tonight is some of the things that we're doing at the university to establish this coral as a model system for studying coral symbiosis and especially its response to climate change. And so the first thing that I'll show you is that we are working on growing it in culture. And what that means is that we have to be able to spawn it and breed it in captivity. And yes, indeed, not only are corals animals, but they have sex. And the sexual reproduction in corals is absolutely fascinating. The most special thing about coral reproduction is that typically it happens only in a very, very narrow window throughout the year. In the case of Astrangia, it happens over about a 2-week period in the late summer. And I'll show you a video of how it happens. So if you take a look at the life cycle here that one of my students illustrated for us, this is to represent an adult colony. And a single adult colony will release either eggs or sperm by the millions. And those eggs and sperm will then meet in the seawater. And we call this external fertilization in the marine biology world. That fertilization will happen in the seawater, and then embryonic development will proceed, and those embryos will eventually develop into small, what sort of look like little fleas kind of dancing around in the seawater. And those are larvae, and those larvae will swim around in the seawater until they find a suitable place to attach and settle onto a rock. And then once they've done that, they go through metamorphosis and then further asexual reproduction to build what we know as a coral colony. And so our goal was to basically conquer each one of these steps in the Astrangia life cycle to get a full, what we call, to close the loop and get a full life cycle in captivity. And so here's where we are on that. So these are here on the left. This is an underwater shot of some colonies that we have in the lab. And what I didn't mention yet is that these corals are very, very small. So unlike the big branching corals of the tropics, and fittingly, if this becomes our state coral, these corals are very, very small, right? So they are the smallest of most, not quite the smallest, but they're really, really small compared to most corals, I'd say, about the size of a silver dollar. And so what you're seeing here is coral colonies that are attached onto little ceramic pucks in our systems. Here is a single colony, and if you look really closely, you might see little specks that look like little specks of dust, and those are eggs that this colony has just released. And what I want to show you now is a video that one of my students took, and this was taken by an otoscope. So if you can imagine a camera that's designed to go into a human ear, we actually can put it underwater. They're waterproof. And so we can get extreme close-ups. And what you're looking at here is a single coral animal, and this is what's called a coral polyp. Here's another polyp over here, another polyp. And the polyp is basically a circle of tentacles surrounding a mouth. And it's through the mouth that the eggs are released. And it all happens within a moment. There they go. So those are eggs that are being released by a single colony. This is obviously a female colony. And so what we can do is we've figured out sort of how to reliably get them to release, when to collect them, and then to get them to release in lab. And then we can mix eggs and sperm together to do fertilization in the lab. And so one of my students did a great embryology study taking pictures of these embryos as they develop. And when they get to a certain stage, they basically have plenty of structures that allow them to swim around in the seawater, and we can hold them in culture tanks at the university. And this is in collaboration with the shellfish aquaculture facility that's on the campus. And it turns out that because we have the ability to grow in really, really large batch cultures, this was designed for raising oysters. We can grow lots and lots of algae. We can feed these larvae as they're growing, and continue to hold them until we can figure out how to settle them. Now, the last bit of that life cycle is where we've been hung up. So what you're seeing here is a brand new baby coral that has settled on a rock. And this was a rock that we actually collected in the wild. So we can see that they do what's called recruit in the wild. They settle in the wild. But we have not yet predictably settled them in the lab. We've gotten some settlement in the lab, but it's not necessarily on the things we've tried to settle them on. So we're still working on that last bit of the life cycle, which is right here. And so the aim is to basically get them in culture so that we can start with a single parent colony and get dozens, if not hundreds or more from a single colony. This is part of sustainable culture, so that as we're working with this organism, we're not overtaking from wild populations. The other thing that we're doing in the lab is we're working on asexual propagation of adult corals in the lab. And so we collect adults, and basically what we can do now is take a single adult colony, which again is about, this big, cut it into quarters, and we can watch it continue to sort of heal its wounds and then grow again. And so in that way, we can propagate corals and again, reduce the amount of take that we have from wild populations. And I have students who are working on these different methods in the lab and photo documenting growth, so we can sort of estimate how quickly we can grow them and which conditions are sort of the optimal ones for allowing this growth to occur in captivity. So those are two of the things that we're doing in the lab to sort of get growth and culture going, so we can establish this as really as a site for providing stocks for ongoing research. And what I want to do now is just tell you a quick story. It's a bacterial story. So I am a coral biologist, but I'm also a microbiologist at heart. And which you probably could tell from my what going on about yogurt at the beginning here. So what does yogurt actually have to do with corals? Well, it turns out that bacteria are absolutely critical for the health of corals. And really ask any microbiologist, and I think we'd all say the same thing, that they're critical for everything. And in fact, they run the world. But back to corals, I would say the thing that's been most interesting to me is thinking about what kinds of microbes live on these corals, how that compares to microbes that are on tropical corals, and what that can tell us. And so what I didn't tell you, I said that Estrange is good at handling the winter, but it does the same thing that we all do, right? It huddles up. And in fact, it actually goes into a dormancy state in the wintertime. And that dormancy is something that we call quiescence. And so once the temperature drops below a critical threshold, those corals actually bring their tentacles in, and they sort of puff up the disc that covers the rest of their bodies. And they stay tight in there. They don't feed. Their algae don't make any sugars for them as far as we can detect. There's basically very, very little activity happening. And we suspect that this is a period where they're sort of regenerating energy stores for reemerging in the spring. And in the spring and the summer, they feed, they photosynthesize, or their algae will photosynthesize or basically use light to make sugars to feed the coral. But what is really neat is that there's a sort of when we did an analysis of the microbes that are in these corals, we could see a very distinct signature. And it was so stable that we called it a core of bacteria that seemed to be associated with Estrangia in the fall. And they appear to be throughout the entire year. They're present, whether it's fall, winter, spring, or summer. And then each season there's a little bit of variation, but the most striking difference that occurs is that in the winter corals bear the signature of diseased tropical corals. So I'll say that again. So the winter corals look like diseased tropical corals from the perspective of microbial signatures. And then what's even more interesting is that in the spring, as they reemerge and they come out of that dormancy, the corals appear to reset, and they sort of reset to that core signature that looks like a healthy coral. And it looks like what Estrangia looks like through most of the year. And so this is work that was done by now Dr. Allison Kerwin, and we published this a few years ago, but it got us thinking about whether or not we could use this really as a model to think about what does it take to go from disturbed disease state back to recovery. So can we take a look at what's going on in this coral from the end of winter or even beginning of winter until spring? And can we model in a coral what's happening that allows that coral to come back from being diseased? And so we're now really interested in sort of doing more to pursue what it is that happens to make that recovery possible. And we've done some laboratory experiments where we've hit the corals with antibiotics to sort of mimic another type of disturbance and then watch them recover naturally. And we're getting new new ideas that I hope to be able to share soon about which bacteria are really important for sort of driving that recovery and maybe saving the corals, um, and sort of helping them to be more resilient. Another piece of that is also looking at bacteria that live on the surface of the corals. And so this is something that we know from tropical corals, that in general, bacteria are coated with a surface, I mean, with a, well, they're coated with a mucus layer on their surface. And you can think of this kind of like the surface of our GI tract, our intestines. So also coated with mucus, which is inhabited by bacteria. And so again, that same analogy applies. If the good ones are on there, we have great digestion, great health. If the bad ones are on there, potentially disease. And so one of the first lines of defense for corals is that they harbor a lot of bacteria that produce antibiotics that prevent infection. And so they protect them from pathogens in the seawater. And so when we first started working on this, no one had really looked for the potential for antibiotics being produced by bacteria on the surface of this coral. And what we were able to find, again, this is work done by my undergraduate students, we took some corals from the wild and used them to grow up bacteria on plates in the lab. And then we isolated those bacteria into pure, what we call pure cultures. And we made a library of those pure cultures, and that looks something like this. So this is basically, each one of these rectangles is an agar plate that holds, let's see if I can zoom in on this, that holds a grid. Each one of these specks is a different bacterial culture. And so we're forming libraries in the lab that are all, originally they came from the surface of this coral. And so I have students who are maintaining these libraries in the lab, and then we're using them to see if we can find evidence that they produce antibiotics, that they can basically kill other bacteria. And so what we do is we test them against bacteria that we've grown from seawater. And so what you're looking at here is a single agar plate, just a culture plate. It looks a little bit cloudier than the previous ones because it has what we call a lawn of bacteria growing across the surface. And so that bacterium is just a single pure isolate that we've grown from seawater. And then onto that lawn, we used a tool called a stamp. Actually, I'll go back to this picture just for a second. So this is one of my students using this stamp. It's basically got 96 little spots that we can hold up to a flame to sterilize it and clean it, and then dip it into a culture, 96 individual cultures of bacteria, stamp it onto a plate. And so when we do that, the bacteria will grow up. And wherever you see a spot here, we've had bacteria grow up. Now, what you're seeing in these two areas here, I'll blow up one of these, is that you have the bacterial, and these 96, each one of these dots is from the surface of Astrangia. So this bacterium here that came from Astrangia is causing what we call a zone of inhibition. And basically what that means is that it's preventing the growth of this seawater bacterium, this tester strain that we had in the lawn. And so overall, what we see in Astrangia is that we can find a lot of bacteria that cause that zone, that killing zone, that prevent growth of bacteria from seawater. And right now, these are the different bacteria that we've isolated from Astrangia that appear to have antibacterial properties. The other thing that's really exciting is we now tested them against a pathogen that we know is responsible for killing shellfish, including oysters and shrimp. It also kills corals, and we've tested it. It actually kills Astrangia. Most of these are active against that as well. So the applications for the aquaculture industry, not to mention human health, are potentially really promising. So that's another thing we're really excited about with this coral. And so the final study, so I'm sort of giving you a few different vignettes here, but the last study that I just want to mention is that we're using Astrangia to study microplastics pollution in a different way than I think a lot of people are, especially with corals. And we're taking advantage of the fact that Astrangia itself is a really aggressive, voracious eater. So what you're looking at here, this is sort of the, well, this is a single polyp, right? Each one of these is a tentacle surrounding the mouth here. And this is an Astrangia polyp that has been fed in the laboratory polyethylene microbeads. And so you can see one that's going to get sucked right into the mouth of the coral. And so we can use these microbeads in the lab and treat them with different things to try and learn more about what influences the dynamics of accidental ingestion of microplastics. And we can see the effects of exposure to different levels of microplastics on these corals. And so, really the main question that's driving what we do is how does microplastics ingestion by corals impact their health? And so I have a team of researchers who are working on these questions as well. And one of the main things that we found, or actually, I guess I should say the first part of the story that really was surprising and troublesome to find was that we looked at Astrangia first in the wild just to see how much microplastics are they coming into contact with. And I'm sure you all have heard that microplastics pollution is a problem globally. And I can tell you that it's certainly very easy to detect them in Narragansett Bay. And so at a site where we work with Astrangia here in Narragansett Bay, the top is showing you a piece of an Astrangia sample that has been ground up in the lab. Everything that you see here that looks sort of like a fluorescent fiber is a piece of microplastics that was found in the sample. And so when we analyzed coral colonies, what we found was that overall there were more than 100 pieces on average per polyp in those corals. So each one of those mouths had ingested over 100 particles on average. And we broke it down to say, well, how many of them were microbeads, how many of them were fibers, and how many of them were other sort of more rough or hard to categorize shapes. And the majority of them were these microfibers that we think are most likely from things like monofilament, from laundry lint, and clothing, wastewater processing effluent. Another important part of um, microplastics toxicity to animals in the marine environment is how they affect their feeding. And so what we found when we did again work with these beads in the lab, so here's an Astrangia polyp, and these blue beads that you see here are the polyethylene, the plastic microbeads, and then right here in orange is a brine shrimp egg. And so when we presented the corals with equal amounts of brine shrimp egg and microplastics, over and over again, they significantly took up more plastics than they did their natural food, these brine shrimp eggs. And so one thing that provides literally zero nutritional value was taken up more than something that is a natural food that has lots of great proteins and fats and are a delicious snack for a coral. And so then we thought, well, it's okay because what we see consistently is that they take these beads in, but then they actually spit them back out. So not all hope is lost. Well, it turns out the other piece of the story is that once they've taken in the beads and they've spit them out, their subsequent feeding is depressed. So it's a tough story. The take home right now is that when corals like Astrangia are presented with microplastics in the water, it does change their feeding behavior. And that's an expensive process, by the way, especially for a marine invertebrate to eat something and to spit it back out. And that's likely why then they feed less as a result after they've gone through that process. One caveat that I'll say before you decide to eliminate all seafood from your diet, I can tell you that oysters are much more selective feeders than Astrangia, and I think that's an important thing to be considering as we think about the impacts of microplastics pollution in the environment. Is that oysters have been shown to actually be able to detect microplastics and kick them out before they filter them into their bodies. And so The last part of this microplastic story that I want to mention that I think is on a lot of microbiologists' minds is this, is that bacteria also have been shown to grow on microplastics. We know that bacteria basically will colonize any surface in the marine environment. And so even if it is polyethylene or some other microplastic, there are certain bacteria that will grow on those microplastics. And when we mimic that in the lab, what we can do is take these microspheres and we can coat them with bacteria that are fluorescently labeled. And what we see is that when they take up those beads, they take up the bacteria basically go along for the ride. And so here's a looking down at a coral, right? Hopefully you can see the tentacles here. Here's the mouth opening. The blue represents the beads that we fed them. And the green is green fluorescently labeled E. coli cells. And so they definitely go into the coral as sort of incidental riders, right? And so we're really interested in what the physiological consequence is. When there are organisms that accidentally ingest microplastics, and those microplastics have unique bacteria on them. And that's something that's been shown over and over again, is that microplastics have very different bacteria on them than other particles that are in the seawater. So if they take them in, what bacteria are they taking in? And one thing my lab is focusing on now is once they spit them back out, who sticks around, who gets sloughed off and stays behind inside of the animal? So again, what are the consequences to the animal's health with running into these microplastics? So those are the different questions that we're addressing in my lab. And I want to say that, one of the things that I think about a lot as a research scientist in this era where we have a lot of really big problems to tackle, is that the most important thing about doing research and about trying to apply that to conservation and doing good in the world is to do it as a community effort. Um, and of course, I think your organization is a perfect representation of that. And in science, it looks like this: conferences, right? And so I would certainly never say that Astrangia research is only done in one place. And I'm really proud to say that at the beginning, when I first started working on Astrangia, there were really just a handful of us who were sort of engaged in doing Astrangia research. Um, it's worth mentioning that Astrangia, the first known description of the coral itself, was in Rhode Island, in Castle Hill in Newport, in the late 1800s by a famous naturalist out of Harvard. His name is Agassiz. And then fast forward about 50 to 70 years, there was some research done on Astrangia again. And then fast forward sort of a lull for Astrangia research. And then some of the most important foundational research on Astrangia occurred at the University of Rhode Island in Dr. Mike Pilson's lab in the 70s and the 80s. And it sort of remained this like really quiet, small, hidden gem, and the research is just amazing. Um, and there have been people sort of independently doing Astrangia research since then. But it was really in 2010 that my colleague and I from my colleague from BU, Dr. Randy Rojan, she and I started talking about getting some people together to work on this fascinating coral because we thought it had so much potential to answer questions that we just couldn't answer in tropical corals. And in 2016, we met another researcher, Dr. Sean Grace from Southern Connecticut State. And the three of us co-founded what we now call the Astrangia Research Collaborative. And in 2016, you can see that we were a relatively small group, and the people keep getting smaller in the pictures, which makes us very proud because the group keeps getting bigger and bigger. And we now basically, and by the way, you can imagine why there's no 2020 gathering photo, unfortunately. This year we'll probably have a virtual conference, but we did miss getting together last year. This group is now not even just a group of New England coral researchers, but coral researchers from all over the country who have been studying tropical corals but are recognizing the value of this coral. And so we were able to basically start this collaborative and just let it grow. And we still, I'd say once a month, a new researcher or a new educator contacts one of us and says, hey, I just heard about this coral. This is really neat. I want to be part of this group. And so if there's someone out here tonight who's a researcher or an educator, we're very interested in moving a lot of Estrangia science into K through 12 curriculum, into public outreach and education. I have the website here. So the website is housed at the Boston University website. And there's a lot of information there about the group, about when we meet, and communication that we maintain, and different experiments that are going on. And finally, I just would like to say, so this is a clip from the article. I mainly have this picture in here because I want to show you just how small these corals really are. But I hope that what I've done tonight is convince you that there's some really neat research and research potential that can be unlocked by working with this unique coral. We're doing a lot of that research here at Roger Williams, and tons of collaborators all over the country are using this coral to answer what are increasingly urgent questions about corals and ocean health. And so with that, I'll thank you for your time, and just put this slide up to show some important collaborators and funding sources, and I'm happy to take any questions.